right, guys, today we're going to be doing a review of some chapter 4 and a preview of chapters 10 and 12. So this is going to be a little bit different from what we've been talking about. So we're moving away from the skeletal system and moving sort of back to talking about cells and their plasma membrane and membrane transport. And then we're going to apply that to resting membrane potential, which is a concept we kind of glossed over. It didn't really talk much about when we covered chapter 4 the first time. And then I'm going to try to just apply this a little bit so that when we get to chapters 10 and 12, you guys will remember it. This is also important because on our upcoming exam, you're going to have a few questions, 2 questions from chapter 4, and they will likely come from this material. All right. So remember, membrane transport is pretty complicated, and it's divided into 2 general types of processes. Passive processes that do not require any extra cellular energy, and during these passive processes, substances move down their concentration gradient. And then active processes, which do require an expenditure of cellular energy. And so when this happens, usually substances are moving up their concentration gradient or against their concentration gradient, or we're forming a transport vesicle, right? And to move a lot or big amounts of substances. And we'll talk a little bit about that at the very end today. So we're going to focus just a little bit on mainly channel-mediated facilitated diffusion, primary active transport, and then a little bit about exocytosis. Those are sort of the 3 main ones that we really have to review and really know well for moving into the skeletal, for moving into the muscular system and the nervous system. So remember, once again, passive processes do not require energy, and they depend on substances moving down their concentration gradient. And that means they're moving from an area of more substance to an area of less. And remember, this ties into one of our key concepts of physiology, which is flow down gradients. So in general, things are going to move from an area of high concentration to an area of low concentration. Things will move from an area of high heat to an area of low heat. And things move from areas of high pressure to areas of low pressure. So that's something that we'll see repeated again and again in physiology. The 2 types that we discussed previously are diffusion and osmosis. Osmosis is really important, which is the movement of water, but we're not going to talk about that much today for this review because we're specifically going to be targeting topics that are going to be important for our nerve and muscle conduction. And so we're going to focus on diffusion. Now for our active processes, once again, they require energy. Substances must be moved up or against their concentration gradient, or we have formed a membrane-bound vesicle. And with that membrane-bound vesicle, it doesn't really matter in which direction the substances move. Although often, because there's a lot of substance, it'll often be down the concentration gradient. But it's more about we need the energy to form this whole vesicle and make this, move the vesicle around the cell. And so usually this is for releasing bulk or big products like hormones or neurotransmitters, right? Which is why it's going to be really important for our nerve and muscle conduction. So we'll start with diffusion, and remember there's 2 main types, simple diffusion and facilitated diffusion. And so with simple diffusion, molecules can move unassisted between the phospholipid bilayer, right? And this is going to be small and non-polar solutes, the ones that are really good examples that we've talked about a lot are oxygen and carbon dioxide, but also some fatty acids, ethanol and urea are other good examples. And we've had a couple of short answer scenarios where you had to talk about this. These cannot be regulated by the plasma membrane, right? Because the plasma membrane cannot prevent them from moving through. They're always going to just follow their concentration gradient. They will move as long as the concentration gradient exists. And so here's an example. We have our phospholipid bilayer with our polar heads and our nonpolar tails. This nonpolar area here, this is that hydrophobic core to the phospholipid bilayer that repels a lot of charged molecules, larger charged molecules or ions. But so oxygen and carbon dioxide are small and nonpolar, and they can move directly through. This will be really important concepts in the respiratory system, which is another reason I'm kind of reviewing it here. So now some molecules are either polar, right? They have partial positive and partial negative charges. They're larger and polar, or they're ions, right? Where they're ions, so they have a full positive or negative charge. And those are going to be repelled by that the hydrophobic, inner layer of our plasma membrane. And so these cannot move directly through the plasma membrane, and they require assistance from plasma membrane proteins, or integral proteins. They're the ones that go all the way through from one side of the membrane to the other. And so we have two types, carrier-mediated diffusion or channel-mediated diffusion. And so carrier-mediated diffusion is when a small polar molecule is assisted across the membrane by a carrier protein. And so it's going to change the shape of the protein binds the molecule, it changes the shape, and it kind of spits it out on the other side. We'll talk about this more, especially in the kidneys chapters. But for review purposes this week, we're going to be focusing on channel-mediated diffusion because we're going to be talking a lot about different types of channels and where they're found on the nerve and the muscle cell. And so this is going to be a movement of small ions through water-filled protein channels. And each channel is going to be specific for a specific type of ion. And so the channel proteins allow ions to move down their concentration gradients, right? Remember, this is a type of passive transport, so the concentration gradient is going to determine whether the ion moves into the cell or out of the cell. So it's all based on that concentration gradient. And so when these channels are open, they allow a specific type of ion to diffuse. And they are specific for one ion type. Now, there are a lot of different types of channels, so we can categorize them into a few different areas. And the first type is a leak channel. And as the name implies, they are always open. They are always open for continuous leakage or diffusion. Once again, it's all passive, so anything diffusing through those leak channels is going to be going down its concentration gradient. And so here's an example, and we'll talk about these a lot. So we have sodium leak channels and potassium leak channels. And so these leak channels are always open, and they just allow one specific type. So the sodium leak channel only allows sodium through, and the potassium leak channel only allows potassium through. And it's going to allow them to move down their concentration gradient, in whichever direction that is. So if we have more sodium outside the cell, sodium will try to move into the cell. If we have more potassium inside the cell, potassium will try to move out of the cell. Remember this, we're going to come back to it. Now, these are useful, but we can't really open and close them, so we can't really control the movement as well. Control the movement of these ions. So in order for the cell to be able to control the movement of these ions, we now have channels that have a door that can open and close, or a gate that can open and close. And these are called gated channels. Gated channels are usually closed, but they open in response to a stimulus for a fraction of a second before they close again. And so types of gated channels, and here are the three main gated channels, are chemically gated channels. We can also call these ligand gated channels. And they are normally closed, but they open with response to the binding of a chemical, like a neurotransmitter or hormone. In this case, we're talking about neurotransmitters in chapter 10 and 12. And so these are going to respond to the binding of a chemical, and so we call them chemically gated channels. And so when the specific chemical that fits that receptor, when it binds, it's going to open for a second and then close again. Voltage gated channels are normally closed, but they open when the membrane changes charge. So we'll talk a little bit about the membrane and how it usually has a charge in it. So there's a charge difference between the inside of the cell and the outside of the cell. And when that charge difference changes, when the voltage changes, then that can trigger the opening of the voltage gated channels. Once again, they open for a short amount of time and then close again. And then finally, we have modality gated channels. And so these are just gated channels that are normally closed, but they open in response to some other type of stimuli that's not a chemical or voltage. And so a good example of this would be like a stretch receptor, right? So it's going to open in response to the stretch of the cell. These are often found in membranes of sensory neurons that respond to changes in their environment. And we'll talk more about those in chapter 16. So the two we're really focusing on here are chemically gated channels and voltage gated channels. All right, so that was passive transport. Now we're going to do a little review of active transport. And so we're going to talk about active transport first, which is going to be active transport without vesicles first. And then we'll move into the vesicular transport at the very end of this. So active transport without vesicles is going to be the movement of a solute against its concentration gradient or up its concentration gradient. And so this is from an area of lower concentration to higher concentration. So we're pushing that bowling ball up the hill, right? It's hard to do because these solutes, you know, they want to be at equilibrium. They want to be distributed evenly. They don't want to all be shoved together in one area. So it takes energy to do this. These are important because it maintains a gradient between the cell and the interstitial fluid. So let's say maybe the cell would like to have all the potassium inside, and so it's going to actively transport all the potassium inside. And so then there's a lot of potassium inside the cell and not very much outside of the cell. That's what we call a gradient. Remember, a concentration gradient, where we have a difference in concentration from on one side of the membrane versus the next. Compared to the other side of the membrane. So the source of energy determines whether movement is primary or secondary active transport. Primary active transport is going to be the one where ATP is the source of energy, and that's the one that we are reviewing today. Secondary active transport, if you remember, is when we use the movement of one solute down its concentration gradient to power the movement of another solute up its concentration gradient, which is also a very important type of active transport, but we're not going to be talking as much about it in chapters 10 and 12. So that'll be more like in the kidney chapters later. So we're not going to review that right now. We're going to focus on the really relevant topics for the next chapter. So primary active transport is going to use energy directly from the breakdown of ATP. And so the phosphorylation of the carrier occurs. And so breakdown of ATP results in a phosphate group added to the transport protein. And so the phosphate that was on ATP is going to break off and bind to that transport protein. That is going to change the protein shape and result in movement of the substance across the membrane. So remember that a lot of times when things bind, when something binds to a protein, it can cause the shape to change. And the shape is really important for the function. And so whenever the ATP, that phosphate, is going to then bind to the transport protein, which is going to change the shape of the protein, and it's going to cause that the pump to work. These are really important because they help maintain concentration gradients by moving substance against their concentration. So the most common types of primary active transport is going to be an ion pump, and this is what we're going to be talking about a lot in the next couple of chapters, which is why I'm focusing on it here. So ion pumps are cellular protein pumps that move ions across the membrane. They maintain internal concentrations of ions. This is how the cell can decide, what to keep inside and what to pump out. For example, this is a red blood cell and this is a calcium pump. So calcium pumps in the plasma membrane of red blood cells, or erythrocytes, are going to be pumping calcium. They're going to prevent the cell rigidity from accumulated calcium. So we're pumping calcium out of the cytosol. So see how, here's the cytosol, this is the inside of the cell. So when the ATP binds, right, so that phosphate's going to bind here, and there's going to be a little calcium in here, and when it binds, it's going to cause this to change shape, and it's going to spit the calcium out on the other side. So the energy from ATP is used to pump the calcium up its concentration gradient outside of the cell. If we have too much calcium in one spot, it can start calcifying and things and cause the red blood cells to become rigid, and we don't want that. They need to be really flexible so they can get through those really tiny blood vessels. And so by pumping the calcium out, it lets the red blood cells, or erythrocytes, remain flexible enough to move and to get through those little capillaries. Another really, really, really important type of pump is called the sodium-potassium pump, or the NAK pump. This is actually a type of exchange pump. It moves one type of ion into the cell against its concentration gradient, while moving another type of ion out of the cell against its concentration gradient. And the plasma membrane preserves these steep gradient differences. So this cell, specifically the sodium-potassium pump, continuously exports 3 sodium outside of the cell and moves 2 potassium into the cell all of the time. So it's going to be important to know these numbers, especially when we get to our next couple of chapters. And so here is the picture up here of our sodium potassium pump. Once again, it's primary active transport, so ATP is going to be bound. So first we have these 3 sodiums here that have gotten, they're sitting in this nice little area inside this protein. The ATP binds to the binding site, which causes the protein to change shape, and it kind of kicks those sodiums out. Then that potassium is then going to go into its little binding sites on the other side, and it's going to bring the potassium inside the cell. And so we have 3 sodium are pumped out of the cell, and 2 potassium are pumped back into the cell. And this is going to be really important for maintaining what we call a resting membrane potential, and we'll get to that pretty soon. So these are some important pumps and channels that we're going to find on the plasma membrane, especially of our muscle cells and our nerve cells. And so the entire plasma membrane is going to have leak channels. So we said leak channels are those ones that are always open. They don't have a door or a gate. They're always open. So there are going to be many potassium leak channels, and there are fewer sodium leak channels. And then we also have sodium-potassium pumps. So as you can see, sodium and potassium are 2 really important ions that we're going to be talking about. These leak channels and sodium-potassium pumps work together to maintain what's called a resting membrane potential. And that's what we're getting into in this lecture. So the resting membrane potential is established by the plasma membrane, and it maintains an electrochemical gradient. Right? So electrical, there's going to be a change in charge, and chemical, there's going to be a chemical gradient. So we'll have both an electrical and a chemical gradient across the plasma membrane. And this is happening when cells are at rest, so we call it the resting membrane potential. This is essential for nerve and muscle cell function. And so that's why we're reviewing this now. So some characteristics of the resting membrane potential. So things that we need to have. One is that there is an unequal relative amount of positive and negative charges across the cell, and that's the electrical gradient we talked about. And so the inside of the cell is more negative compared to the outside, and the outside of the cell is comparatively more positive. And you can measure this with a little, like if you put little electrodes inside and out of the cell. This unequal distribution is a type of potential energy, right? Remember, opposites attract. Negative charges are attracted to positive charges and vice versa. And so we have this difference where we have a lot of negative on one side and positive on the other, but there's a barrier in between and they can't move, right? And that's a type of potential energy. So the other characteristic of resting membrane potential is that we have ions that are unevenly distributed across the plasma membrane due to the actions of ion pumps. And so this is called a chemical gradient. So that's the chemical part of the electrochemical gradient. And so inside the cell, we have a higher concentration of potassium in the cytosol versus the interstitial fluid, which is the outside of the cell. So we have more potassium inside the cell. And then outside of the cell, we have higher concentrations of sodium, chloride, and calcium. So inside the cell, we have more potassium. Outside the cell, we have more sodium, chloride, and calcium. And this unequal distribution is also a form of potential energy. So ions are going to want to move down their concentration gradients, but are prevented from doing so by the plasma membrane barrier. And so we talked a little bit about potential and kinetic energy in chapter 3, and I wanted to bring this picture back from chapter 3. And there is potential energy in the plasma membrane. And so this plasma membrane is a barrier, and it's a barrier that unless there is a channel in there, a lot of times it's going to prevent the movement of ions, especially from one side to the other. And so if we have a concentration gradient that exists across the plasma membrane, then let's say we have more sodiums on the outside of the cell and less on the inside of the cell, the sodium really wants to move from the outside of the cell to the inside of the cell, but it can't. And so that energy of like position, that's the potential energy, where the sodium wants to get in the cell, but it can't. It's like building up that potential energy. And so if this channel is closed, it's potential energy. Right here, it's building up. But when we open this channel, that potential energy can be converted to kinetic energy. So that sodium is going to be like, oh, there's an opening, and then it goes right down its concentration gradient. And so the movement of that potassium or excuse me, the movement of that sodium down its concentration gradient is going to be that kinetic energy. Remember, kinetic energy is the energy of movement. And specifically, when we have the movement of charged particles, like ions, we call this electrical energy. So this is going to be electrical energy inside our body. And so our neurons and our muscle cells are both going to have these electrical sort of charges running up and down their plasma membranes. So, but in order for any of this to happen, we first have to establish and maintain our resting membrane potential. And so our resting membrane potential, the most important ions that we'll talk about in resting membrane potential are sodium and potassium. And the movement of sodium and potassium depends on the electrochemical gradient, remember, which has an electrical portion and a chemical portion. So first we'll talk a little bit about potassium. Potassium is actually the most important determinant of the specific value of resting membrane potential. And each different type of cell is going to have a slightly different sort of average resting membrane potential, and we'll talk about those in a little bit. Potassium is going to move down the steep concentration gradient through potassium leak channels. And remember, we said there are relatively a lot of potassium leak channels compared to sodium leak channels. And so potassium is going to go from the cytosol to the interstitial fluid. Remember that when we pump our sodium potassium pump that is setting up this resting membrane potential is pumping potassium into the cell. So potassium is going to want to move out. I'm going to pull up my whiteboard and talk about it here. All right, so here's my beautiful whiteboard. This right here, this is going to be the plasma membrane, and in this little area, I drew in our phospholipids here. There's some cholesterol in there, but here I just kind of graded out because I wasn't going to draw all those. So this is our plasma membrane. And remember, we said on the entire plasma membrane, we're going to have potassium leak channels, sodium leak channels, potassium leak channels, potassium leak channels, and we're going to have a sodium potassium pump. And our sodium potassium pump is going to pump 3 sodium outside the cell and 2 potassium inside the cell, and it's going to need ATP. And this happens a lot, all the time. So what I want to do is add some more potassiums. And so we can look here and say, oh, well, their potassium is always being pumped in. So where are we going to have more potassium? We'll have more potassium on the inside of the cell, right? So we're going to we have a bunch of potassium on the inside of the cell. Remember, so this is because we have a lot of these sodium potassium pumps, and so we're constantly going to be pumping potassium into the cell. And then, so if we have a lot of potassium inside the cell, where do you think potassium is going to want to go with these leak channels? Right, so potassium is going to go, it's going to want to escape, right? It's going to want to go down its concentration gradient. So our potassiums, some of them are going to want to go down their concentration gradient through the leak channels. And so some of them will diffuse down the concentration gradient. So I want to look at potassium. And so chemically, here we have potassium. And so chemically, where is potassium going to want to go? This is the inside of the cell here. This is inside of the cell. This is outside of the cell. So chemically, we have a really high concentration of potassium inside the cell. And we have a very, very we just got 1 or 2 potassiums outside the cell, not very many. So chemically, where does potassium want to go? Right, potassium is going to want to go outside the cell, chemically. All right, so we're going to fill out this chart as we go. The other thing, though, that we're looking at is electrically. And so did you notice down here that we have these little, these are going to be proteins. And so these are negatively charged proteins, and they have negative charges over here. And so some of our potassiums, right? What are potassiums? Are they positive or negative? Right, potassiums are going to be positive, right? So these little positive potassiums here, some of them aren't going to want to leave, right? Because they're going to want to stick over here with these negative cells. So electrically, the inside of the cell is more negative than the outside of the cell. So electrically, where is potassium going to want to go? Remember, opposites attract. Potassium is positive, So is it gonna stay in the positive area outside? Is it gonna want to go outside for the positive area, or is it gonna want to stay in the negative inside of the cell? Remember, opposites attract. So potassium is kind of electrically gonna want to stay in, right? So some of these guys up here are gonna diffuse out down the concentration gradient, but some of them are kind of gonna want to stay in. Potassium's a little bit more conflicted. So it's electrically, it wants to move down, or excuse me, chemically it wants to move down its concentration gradient to the outside of the cell, but electrically it kind of wants to stay in, right? Because it is a positive cation and it wants to stay in the negatively charged interior of the cell. So potassium is a little conflicted. All right, so let's go back to potassium here. Let's go back to our... So the most important determinant in specific value of resting membrane potential is potassium. Remember, potassium wants to move down the steep concentration gradient through the leak channels from inside of the cell to the outside of the cell. Cytosol is the inside of the cell, interstitial fluid is the outside of the cell. So normally potassium wants to go out of those leak channels. But there are some of those big negatively charged proteins that remain inside the cell, and those negatively charged proteins are also pulling on the potassium. And so remember, it's acted on by both the electro and the chemical parts of those gradients. So the positive charge outside repels the movement of potassium out. The negative charge on the inside attracts the potassium inside. But at the same time, potassium is moving down a concentration gradient towards the outside of the cell. So like we said before, potassium is a little bit conflicted. So it's going to, some of it will diffuse out down that concentration gradient, but then some of them are going to want to stay in the cell to stay near those positively charged proteins. And so we're going to just have a little bit of diffusion out until equilibrium is reached. All right, sodium is next. And so let's look at sodium on our whiteboard. All right, so sodium, here, we have 3 sodiums here. Let's pick up the blue pen. And so our 3 sodiums here, if we are always pumping sodium outside, where's the sodium going to be? Right, we're going to have a lot of sodium outside. A whole bunch of sodium outside the cell. So we have lots of sodium outside the cell and hardly any on the inside. You know, like 2. There we go. A few sodium on the inside, but really lots of them are on the outside. And so when we have these sodium leak channels, where do you think sodium is going to go? Is it going to want to, which direction is sodium going to go? Right, it's going to go down its concentration gradient. Sodium is going to try to go inside the cell. So chemically, sodium is going to want to go from an area of high concentration to an area of low concentration. Chemically, sodium is going to want to go inside the cell. Now we have to look electrically at what sodium wants to do. Sodium is also a positively charged cation, just like potassium was. So remember, opposites attract. So where is sodium going to want to go? Is it going to want to stay in the positive outside, or is it going to want to go into the negative inside? So since opposites attract, sodium is going to want to go into the negative inside. And so sodium is pretty sure, sodium is not conflicted. Sodium really definitely wants to go through these leak channels. So all the sodiums are going to go through these leak channels, but how many leak channels are there for sodium? Right, there are not very many of them, right? Not that many of them. So some of the sodiums do slide through, and so we do have some sodium that's sliding through, but then eventually the sodium-potassium pump is going to pump it back out. Just like we have some of these potassiums that are sliding through, but then the sodium-potassium pump is going to pump some of those back in too. All right, so once again, sodium is going to diffuse into the cell from the interstitial fluid, the outside, to the cytosol, the inside, at the same time as the potassium's leaving. It's going to enter through those sodium leak channels, but remember, there aren't as many of them. It is being pulled both by its concentration gradient and the electrical gradient. And so there's a lot of sodium wanting to go in, but the leak channels, there aren't as many of them, so we don't have as much sodium entering in. And we're losing more potassium. And so in general, because we're losing more potassium out of those potassium leak channels, because we have more potassium leak channels, And we're gaining some sodium, but we're not gaining as much because our sodium doesn't have as many leak channels to go through. The inside of the cell is going to become more positive, as well as, oh, excuse me, this is a typo. The inside of the cell becomes more negative. And so the inside of the cell becomes more negative, and as well as those negatively charged proteins inside. So the inside of the cell is more negative compared to the outside. And so the sodium-potassium pump is very significant. It maintains the resting membrane potential. So all those ions that are slipping in and out of those leak channels, well, the pump can pump them back to the appropriate sides. So sodium is pumped out, potassium is pumped in, always in opposite directions and always against the concentration gradient. The other thing that I wanted to mention is that a way to remember it is I think of sodium as snakes, right? So do you want snakes in your house? No, you throw them out. So we throw snakes outside. And I think of potassium as puppies. And do you want puppies in your house? Yeah, puppies are pretty cute, right? Potassium is puppies, bring them inside the house. And so we have got, throw the snakes out, the sodiums go outside, potassium, bring them in, the puppies come inside. All right, so this is our resting membrane potential. And now I want to apply it to neurons and muscles. So once again, this is another review. Really, just this slide says the same thing as our previous slides. Ions are unevenly distributed across the plasma membrane due to the actions of pumps. So we have more potassium inside the cell, and then more sodium, chloride, and calcium outside the cell. We haven't talked much about chloride and calcium, but you can just remember that there's more outside the cell. The gated channels are closed at rest. All right, so there are going to be some gated channels. I didn't draw them yet in the membrane, but there are going to be some gated channels that can open and close. But remember that gated channels are closed at rest. I like to think of the gates and gated channels as like gates in a farm. Like if anyone's ever, worked on a farm, gone to a farm, you always close a gate. If you open it, you close it again because you don't want the animals getting out. So the gated channels are closed at rest in general. And there's an electrical charge difference across the membrane. The cytosol, or the inside of the cell, is relatively negative compared to the intracellular fluid. Or, excuse me, the interstitial fluid, which is the outside of the cell. The resting membrane potential is typically negative 70 millivolts in neurons and negative 90 millivolts in muscle cells. You don't have to memorize those numbers yet, but we'll get to them in the next upcoming chapters. And the difference can be measured with microelectrodes, one inside the cell, one outside, and a voltometer. So once again, potassium diffusion is the most important factor in setting up the resting membrane potential. Potassium diffuses out of the cell due to its concentration gradient. But that diffusion out is limited by the electrical gradient, the pull of the negative resting membrane potential on the positive ion. And so remember, potassium is conflicted. Since there are a few sodium leak channels, sodium can also influence the resting membrane potential. Sodium will diffuse in due to its concentration gradient and its electrical gradient. The small sodium leakage means the resting membrane potential in a neuron is around negative 70. So if it was only potassium, if that was the only ion leaked, the resting membrane potential in neurons would be around negative 90. But since we're also gaining some more positives back, we're gaining some more sodium, we have some sodium leakage back in, the resting membrane potential is around negative 70 in neurons. It is actually negative 90 in muscles because muscle cells have a lot of negatively charged proteins and stuff on the inside. Once again, the role of the sodium-potassium pump is very important. By pushing 3 positive charges out and pulling in only 2 positive charges, the pump contributes about negative 3 millivolts to the total. So imagine like if you had a bank account. So the inside of the cell is like your bank account. And so if you spend $3 every day and only make $2 every day, what's going to happen eventually? Right? Your bank account's going to go negative, right? And so we're pumping 3 positive ions out and we're only bringing 2 back in. And so eventually that's going to contribute to some of the negative charge on the inside of the cell. And most importantly, it maintains those concentration gradients for those ions. So we have lots of potassium inside the cell and lots of sodium outside the cell. You'll have to know where those ions are at rest. All right, and so here's a good picture. We've got sodium, chloride, and there's calcium out here too, outside the cell. And inside the cell, we have lots of potassium, as well as these negatively charged proteins. And then we have our potassium leak channels. We have lots of them. We have fewer sodium leach channels, and then we have these sodium potassium pumps here. They're pumping 3 sodiums out and 2 potassiums in. So that is resting membrane potential in a nutshell. Hopefully that was helpful. We're going to be going over this again and again and again this semester because it's pretty important. The last thing I wanted to talk about is going to be vesicular transport, which is a type of active transport. Remember, this is going to be also called bulk transport. And it involves energy input to transport large substances across the plasma membrane by using a transport vesicle, which is a membrane-bound sac that's filled with whatever material it is that you're transporting. And so we can organize it into processes of exocytosis, which is secreting things outside of the cell, and endocytosis, which is taking things into the cell. So for the purposes of this review, it's very specific to what we're doing next. We're only going to be talking about exocytosis because we'll talk a lot about how neurons secrete their neurotransmitters via exocytosis. So remember, exocytosis is when large substances are secreted from the cell. Macromolecules that are too large to be moved across the membrane. Any material that's packed within these intracellular transport vesicles. The transport vesicles are then going to float, well, they're going to be pushed up to the plasma membrane, and then once they hit the plasma membrane, they merge and fuse together because they're both made of that same phospholipid bilayer. And this requires ATP. And then the contents are released outside of the cell following fusion. And so for a good example of this is the release of neurotransmitters from nerve cells. Very important in both neurotransmission and muscle contraction, which is why we're reviewing it here. All right, and so we can see here's our vesicle. It's filled with stuff. It's going to bump into the plasma membrane and sort of merge with the plasma membrane, right? So they become one, and then it releases its secretions outside the cell. And then that transport vesicle is like now merged with the plasma membrane, and it's part of the plasma membrane. And this is how our nerve cells are going to be releasing neurotransmitters, and which will be really important for muscle contraction and nerve conduction. All right, so that is our review of membrane transport, resting membrane potential, and this is sort of a preview. We'll talk a little bit about nerves and muscles in our next unit. All right.